Hello students, today let us discuss about the mutations. So mutations, these are the sudden change in the genetic makeup of an organism, or you can say that they are the spontaneous permanent change in the genetic makeup of an individual. Fine, what are mutations? Mutations, they usually occur in the genetic makeup of the individual, and it is spontaneous, permanent and irreversible. Fine, they are also irreversible. If once occurred, they are not going to get reversed, they are not going to get back to their original one. Let me add irreversible also in the bracket. Let me write irreversible change in the genetic makeup of an individual. So simply we can define them as a sudden change in the genetic material of an organism. Fine. So Dobzhansky, a scientist, he told that the mutations are mistake in the cell division. During the cell division, if something goes wrong with the genetic material, then the change will occur, and those changes are called as mutations. So it is a misprint or mistake which occur during cell division, and due to this we will see the difference in the metabolism. Fine, that we will deal little later. At present we will deal some of the simple and basic things. So what do you mean by mutation? Change in the genetic material. A very simple definition. Fine. And this genetic change in the genetic material is expressed by the organism. Maybe physical, or the change will occur in physical and chemical constitution of an organism. Fine. And the main change will occur in the chromosome or the gene. So first let us see how the mutations will be. Dobzhansky, you have to remember that he told mistake in the cell division. Fine. So mutations, they may be useful, they may be harmful, or they may be neutral. So mutations, useful mutations, they are helpful for the progenies to adjust, to adopt with the existing environment, and they are beneficial, we can say. And harmful mutations, they will not help the organism in adopting with the existing environment. So that is about the harmful. And neutral, the change will occur in the genetic makeup of an individual, but it is not going to affect the organism's survival. And such mutations are called as neutral mutations. They will not interfere with the organism's survival, whereas these two will interfere. One will interfere it in a good way, the other one in a harmful way. And always nature prefers the useful or positive variations, positive mutations which will occur in the organism. Now, mutations are classified into different types based on many things. At present, the mutations which will occur, where they are going to occur, based on that we can classify them into two types, that is chromosomal mutation and gene mutation. Chromosomal mutation and gene mutation. Chromosomes, you already know what do you mean by chromosomes and its structure, you have studied in first year. So the chromosomes are the vehicles of heredity. So in the chromosome, the change may occur in the structure or in the number. If the change will occur in the structure of the chromosome, then it is called morphological change. That is, the physical structure of a chromosome will change due to certain reasons which we are going to deal now. And the next is the change may occur in the number of the chromosomes, the sets of the chromosomes. Those we can say numerical change. Fine. So let us see now the morphological changes which occur in the chromosome. So they are classified depending upon the sequence of genes. If they change, automatically the structure of the chromosome will change. Depending upon how or what kind of change is going to occur in the chromosome, they are classified into 4 to 5 types. 4 types in one only we can say AB. Let me say you. Now the first change is the deletion, or which is also called as deficiency. which is also called as deficiency change. So here you have studied the chromosomal structure. Here a particular segment of the chromosome will get deleted from the original one, and later suppose that here we have taken one chromosome. The sequence of genes, let us take them as A, B, C, D, E, F, G. Fine. So it is a centromere. Suppose C and D will get deleted due to some reason. Maybe the reason may be physical or chemical. That we will deal little later. If this part, this segment of the chromosome will get deleted, these many genes which are present here will get deleted. And here we have taken only two for our convenience to understand. So C, D, if they get deleted, whatever functions were assigned to this portion of genes, this portion of chromosome will be absent in the chromosome which is going to form after this. So thus it will affect the survival of the organism. Fine. So here what is the resulting chromosome will be having? Only it will be having A, B genes, and then it will be having E, F, G. So this arm got affected. So such a kind of changes which will occur in the chromosome due to the loss of a part of the chromosome is called as deletion or deficiency. And this deficient segment will affect the phenotype of the organism. So this is one type, and then let me go to the next one, which is called duplication. So here the name itself says, what do you mean by duplication? Here certain segment of the chromosome will get unnecessarily duplicated. Fine. So that duplicated, so here let me say, what do you mean by duplication? A certain part of the chromosomal segment will get duplicated. That means the genes which are present here will get repeated. Like here I have taken the common example. A, B, C, D, E, F, G only I have taken. So the one which I have marked in the box, B, C, will get duplicated, will get doubled. So in the resulting chromosome, it will be having A, BC is already there. So one more BC, the individual will get, and thus the resulting chromosome will be like this. It will be having the repeated two genes extra. One segment, a part of the segment will be extra there. And this will give that individual additional character. Those character may be useful, harmful or beneficial. One can't say. It depends upon that segment of the chromosome for what it is responsible. Maybe major defect, maybe minor, maybe nothing. Fine, it depends totally on that segment. What work it used to do, that work will be doubled, or it will get affected. Fine. So that is the duplication. Now translocation. Here translocation means between the homologous chromosomes or any two chromosomes, one part of the chromosome will get translocated to the other part of the chromosome. Fine. So from a part of segment of chromosome from one will get attached, will get translocated, will get transferred to the other chromosome unnecessarily, which is not wanted. Fine. So here there is a deficiency and here there is an addition. So such a kind is called translocation. Simply a transfer of chromosomal segment from one chromosome to the other, and it also results in the mutation. Fine. So here I gave the example of A, B, C, D, E, F, G. I have taken here E, F, which I have marked in the box. So these got shifted to this chromosome. Fine. And this segment may get inserted anywhere, maybe before 5, between 5 and 6, between 6 and 7, or maybe after 7. Wherever insertion, wherever it will get in, it can get inserted. So here I have taken before 5. So how the resulting homologous chromosomes will be? A, B, C, D will be there. From here, E, F got deleted. EF got deleted in the sense it got shifted to the another homologous chromosome, another one. So only G is remaining here, and on the other chromosome you will say 1, 2, 3, 4, and this EF got inserted here. So EF, let me write EF, then 5, 6, 7. So this much portion got extra. Fine. So that is about the translocation. The translocation may be inverse also. Let us see that now after this one. So what did I told now? Mutations are the sudden changes which occur in the genetic material of an organism, and Dubinsky told that mutations are the misprint during the cell division. Fine. They may be useful, harmful or neutral, and always nature selects the useful mutations which have got the adaptive value, which will help the organism in survival. And depending on where the change will occur, we can classify mutations into chromosomal mutation and gene mutation. At present we are discussing the chromosomal mutation. In the chromosomal mutation, there are two types. We can classify them for our convenient: morphological and numerical. What do you mean by morphological? Change in the structure of the chromosome. Numerical, change in the number of the chromosome. At present, we have discussed about the morphological changes in the chromosome. Morphologically, four types are there. One more extra, inversion only, we will take reverse one. So here we have discussed deletion, that is a part of chromosomal segment will get deleted, and then duplication, a part of chromosomal segment will get duplicated. Fine. And translocation, a part of chromosomal segment will get transferred to the other chromosome. So thus these changes will result in the mutation. Let me see the fourth one, that is inversion. Now let us see the fourth type, that is inversion. The name itself says inversion, will get inverted. So here ABCD is there. So this portion will get inverted. It will get inverted. It will get rotated through 180 degree. So all the sequences will get reversed. That is why it is called inversion. So here I have taken this segment BCD will get reversed. So DCB. So if this portion will get changed, the coding also will get affected. So that is about the inversion. Now four types we have discussed there. In the translocation, maybe the sequences will get reversed and then get translocated. That one we can add as a fifth type, or as these are the major four types, we have to keep these major four types in our mind. Hope clear. Fine. Now let us go to the numerical changes. That is change in the number of chromosomes. You know that every individual's chromosomal number is fixed and it is not going to change. If change occurs, then the characters of an organism will get affected. So numerical change, let us go through. Here there are two types of numerical changes which occur in the chromosome. That is euploidy and aneuploidy. What are they? Euploidy and aneuploidy. In the euploidy, change will occur in the number of sets of chromosomes. Fine. What do you mean by euploidy? The change will occur in the number of the sets of the chromosome. Number of sets of chromosome. You know that usually haploids and diploids will be there. In diploid organism, two sets of chromosomes will be there. One from the mother side, which are called maternal chromosomes, and other from the father side, which are called maternal chromosomes. That is, paternal and maternal chromosome sets will be there. So diploid, and in the haploid only one set of chromosomes will be there, and these are natural, which are there in the nature. Fine. If by mistake chromosome number will get chromosome sets will get affected, instead of, see, usually if one set is present, then we will say haploidy, two sets diploidy. I told these two are common. Usually in the diploid organisms the gametes are haploid, And triploidy, tetraploidy, pentaploidy, these are seen in the plants and they will play important role in certain plants as they provide resistance to the drought, or they may be having the capacity to tolerate high temperature, maybe disease resistance capacity. That additional characters are due to the ploidy in the plants, but in the animals they will not play a significant role. Let us say if one more set will get added to the diploid, then what do you say for that? Triploidy. If four sets, if that is four sets are present, or you can say to the diploidy, two sets of chromosomes if they get added, then what do you say? Tetraploidy. Likewise, we will go on pentaploidy, hexaploidy, heptaploidy. Like this, if it will be more, the general term what we use is the polyploidy. Polyploidy. It is a general term. That is change or the presence of more than two sets of chromosomes in the individual. What do you mean by polyploidy? Presence of more than two sets of chromosomes in the individual. Depending upon the number, just we will say triploidy, tetraploidy, like that. And all this under what heading it will come? Under euploidy. Now next is the aneuploidy. So here the chromosome number may get added up or may get deleted. Fine. From the diploid set, the chromosome number may get deleted or may get added. Such a kind of change is called aneuploidy. So here let us see when the chromosome will get deleted or added up, what do you say? Monosomy. Mono, single. So from the diploid set, one chromosome will get deleted. Such a change is called monosomy, and this result in disorder, which we will read under genetic disorder little later. In coming period we will read about it. At present, just to remember, if one chromosome will get deleted from the diploid set, then we will see it as a monosomy. If among the diploid set two, both the chromosomes, if they get deleted, then it is called nullisomy. Because here from the diploid set, if one chromosome will get deleted, one chromosome is going to remain. Fine. And here from the diploid set, if both the chromosomes are going to get deleted, nothing will be there. So it is called nullisomy. And to the diploid set, if one chromosome will get added up, then that is called trisomy. One extra chromosome will get added up. Fine. Tetrasomy, here two chromosomes will get added up to the diploid. So it is called tetrasomy. So in the aneuploidy, depending upon how many chromosomes will get added up and deleted to the diploid chromosome. Fine. So they are named into monosomy, nullisomy, trisomy and tetrasomy. And these results in certain abnormality that we are going to deal in coming periods. Now, based on where the mutations will occur, in the organism, the classification can be done into two types, like somatic mutations and germinal mutation. Somatic mutations and germinal mutations. Fine. So somatic, soma, body. Somatic mutations, these are the mutations which will occur in the body cells. So as you know already the meaning, so I did not write that definition. Soma, body. So what do you mean by somatic mutations? The change in the genetic makeup of the change in the genetic makeup will occur in the body cells. Fine. The body cells, in the body cells it will occur, so it will not get inherited to the progeny. It is restricted. It is confined to the organism. It will not get inherited from generation to generation. So it will not play any significant role in the evolution. Fine. So that is about the somatic mutation. And then germinal mutations. What do you mean by germ cells? Reproductive cells. If the change will occur in the reproductive cells, then that will get inherited to the generation. and it will play a very important role in the evolution. If beneficial, then carried on from generation to generation. If it is harmful, then the nature is not going to select it will, and nature will not allow it to reproduce also. You will study this in Darwin's theory. So let me just revise what I told now. I told you the fourth type of chromosomal morphological change, that is the inversion, where a segment will get reversed, that is it will get rotated through 180-degree angle and then get inserted. Fine. So that is inversion. And now numerical changes which will occur in the chromosome. Based on that, we have got euploidy, aneuploidy. In the euploidy, the sets will get added up. Fine. So if single set is present, we will say haploidy, if 2 diploidy, if 3 triploidy, if 4 tetraploidy, if more than 10. Then we will say polyploidy. Usually if more than 2 sets are there, commonly we will use this term polyploidy. And what do you mean by aneuploidy? The chromosomal number may get added up or deleted. If it will get deleted from the diploid set, we will say, depending upon whether it is 1 or 2 chromosomes getting deleted from the diploid. If 1 monosomy, if 2 will get deleted, then it is nullisomy. And based on the addition, if 1 or 2, if 1 will get added to the diploid set, trisomy, if 2 will get added up, it is tetrasomy. And now we are seeing the next method of classification, that is based on the occurrence where the mutation is going to occur. If it is occurring in the body cell, then it is called somatic mutation. If it is going to occur in the germ cell, then it is germinal mutation. Body, that is somatic mutations, are not inherited. They are confined to the organism itself, hence they will not play any important role in the evolution. And germinal mutations will get inherited to the next generation and will play a significant role, or you can say simply a role in the evolution. Let us see the next now, gene or point mutation. So gene or point mutation. Gene, you already know, it is a part which is present on the chromosome and responsible for a particular character, which already you know. And these are called point mutation because it will occur at a particular location on the chromosome. That is why it is also called as point mutation. So the change will occur in the gene, and that too in the sequence of nucleotide. The structure of chromosome, about the gene, genetic code, you'll read in coming topic, that is molecular basis of inheritance. Fine. So what do you mean by gene mutation? The change will occur in the sequence of the nucleotide. Fine. So this is also called as point mutation. And here I have written only one example. Like a gene, it is the sequence of 3 nucleotides together will code for one particular protein. Fine. So here one example I have taken. On the DNA, if A, T, G is there. On the DNA, if A, T, G are there, what are these? You know already. The nitrogen bases, you know, adenine, guanine, thymine, cytosine, and in the RNA, uracil. That you already you know. So on the mRNA, it will be UAC. If the code ATG will be there, it will code for the tyrosine, which is normal. Fine, it is normal. And if change will occur in any one of these codes, it may be neutral, it may be beneficial, harmful, or sometimes it may not code for any protein, or sometimes it may code for the wrong one. So here we have taken one change. Suppose if the change will occur in the G, instead of G, if A will get added, or A will get added in that particular gene, it will code for tyrosine, no problem. Same, the normal protein only is produced, the organism will not get affected. If instead of G, if C will get coded, The protein, it will stop coding tyrosine. Tyrosine is not coded. Then the incomplete protein is formed. Incomplete protein is formed. Suppose if instead of A, G will get coded on the DNA due to the change, due to the mutation, then it will code for the histidine, which is a faulty protein. It will not do any function. Fine. So like this, the gene mutations will get affected. So the one best example we will read in coming period, which is also called sickle cell anemia. Let me not write here, we may get confused later. So we will get this about in next period, where we will read about the genetic disorder. Sickle cell anemia, that is also a best example for the gene mutation. Next we will discuss. Sorry. Now mutation theory, actually I forgot to say you. See, mutation theory for the first time was introduced by a scientist called Hugo de Varese. Hugo de Varese, you already know that he is one among those scientists who rediscovered Mendel's work and published in the name of laws of inheritance, which were proposed by the Mendel. Fine. He is one of the scientists who independently worked and proved that the Mendel is correct. Fine. And this Hugo de Varese, he observed in evening prime rose the appearance of different three characters which were growing wild. When you compare the evening prime rose wild variety and other varieties, there are changes. The changes are many. They used to occur spontaneously, like the shape of the leaves, the size of the leaf, and the height of the plant, the color of the flower, the size or shape of the fruit. Like this, the different changes he observed when he compared the different plants which he grew in his garden. Fine. So then he called these spontaneous changes as mutation. These spontaneous changes as mutation, and the evening primrose, the scientific name is Ornithera lamarckiana. It is Ornithera lamarckiana. Fine. So he contributed to the mutation and he recognized for his mutation theory. It is one of the important contribution which was done by Hugo de Varese, and he also told that the mutation will play a very important role in the evolution. Now till today, whatever we have discussed about the mutation. Again, the mutation is classified based on where it is going to occur, like autosomal mutation, allosomal mutation, based on the type of chromosome, based on types of chromosome. Like if it is occurring, like somatic, what I told, autosomal mutation, allosomal mutation, dominant mutations, recessive mutation. There are many types depending upon the where, how it is going to act, they are classified. So we have discussed the important one which are given in our book. Now let us finish off today this much, mutations. Fine. Tomorrow let us read the pedigree analysis.